Hello, and welcome to our session today, where we will talk about our latest and greatest KingFisher instrument, the KingFisher Apex. This represents for us the pinnacle of automated sample prep. We will cover the basics of how these units work, get a little close-up of the Apex in action, and also go over some of the features that make this a more modern instrument. Following this, we will highlight some of the recent applications that we have developed and maybe cover a few that you are not aware of that the instrument can actually handle. Feel free to go ahead and put any questions as they come up into the chat, and we will have a live Q&A session at the end. So my name is Christian Kiss, and I am a field application staff scientist with our sample prep group at Thermo Scientific. If you saw my previous video on the KingFisher Flex at any time within the last 5 years or so since we made it, I want to personally thank you for taking the time to watch and learn about the product and helping us garner nearly 30,000 views. I could say that we really attracted your attention with that one, and why not? In the end, you probably want your samples to be just like I like my coffee, that's pure, consistent, and ready to fuel great results. So let's dive in. Kingfishers in general have actually been around for quite a while, since 1999 to be exact, with our very first model. After nearly 26 years, the concept has remained the same, but it is now a proven method for automating the purification of nucleic acids and proteins. Along the way, there have been some new models come out to add features, to expand throughput capabilities, and really just enhance that user experience. So let's talk about them real quick. So with the instruments that we have available, you can see they came out in 1999. Now they're not all sold and supported at this time, so we want to focus on the red box units. That is going to be the KingFisher Flex. We still sell it, we still support it. I always say, don't fix it if it ain't broke, right? It's a great instrument, but it is about 17 years old at this point. The additional throughput is going to be with the KingFisher Apex. That's what we're going to talk about mostly today. And with the high throughput, we also have the KingFisher Presto, which is mainly meant to be integrated into liquid handlers, for example, like Hamilton and Tecan, who we partner with on that. If you have low throughput, we also have the KingFisher Duo Prime. With the other instruments, you can do up to 96 or up to 24 samples at a time, depending on your volume format. And with the Duo Prime, that's going to be more like up to 12 samples or up to 6 samples at a time. But it's still a great place to start if you're just getting started with sample purification. Let's talk about the instrument and how it works. To understand that and why they're your best choice, it's always helpful to frame it around some key methodologies to see how they compare. Well, let's start with columns, right? This is basically where most people start because they are easy to acquire and don't really require a lot of other equipment. However, in the big picture, this is not your best option simply because it is a single pass system. Right? Your sample goes through once, your washes go through once, your elution goes through once, and that's pretty much it. There's no shaking going on, no mixing going on, and let's face it, it's not entirely customizable or scalable. It's not like I can pour more column in here if I need to scale up, or cut this in half if I need to scale down. And if you happen to be working with samples that have a lot of particles, well, no doubt you have probably clogged a column or two in your past, as I have as well. As somebody that wants consistency and reproducibility without the headaches, this is just not your best option. So let's get rid of this. All right, from here, we'll move to magnetic bead work, right? And so here we're looking to bind your target to magnetic beads instead of inside a column. And there are a variety of magnets enabled to accomplish this. It's better than columns because you can now add mixing or shaking between the steps. And this, of course, enhances the process. If you don't have a lot of samples, you often do this manually in a liquid movement process. And this is where we attract beads to the bottom or to the side of the vessel and then use pipetting technique to remove and add liquids between the steps. Now, despite this being a better process, the caveat is that when you pipette, it has to be done just right. If your tips get too close to the beads, you end up pulling them out, which of course means loss of your sample. If it's not close enough, well, you're leaving solutions behind that are contaminants, and this can get carried forward until the end. So yeah, if you're thinking about putting this on a liquid handler, it's really the same principle. You still have to make sure that your machine does it just right. So yeah, better, but still not my best option. We're going to get rid of that soon. So the solution then is really using magnetic beads with a bead movement process instead of a liquid movement process. And that is what our KingFisher does. So to describe this process, we've got a nice visual here. We're still going to be using the magnetic beads to bind our target in solution. But rather than pulling those beads down, what we have is a magnet that's on the instrument, and this is going to be covered with a plastic tip comb consumable. This is both to protect your sample as well as the instrument magnet too. Well, that's going to go down and collect all of your beads just on the outer surface and the tip of that tip comb assembly. Those will be transferred to the next solution. The instrument will remove that magnet from the tip comb. So this means your beads are now just resuspended in solution. The instrument will now use that empty tip comb basically as a plunger to do all that mixing. And so you've got this large amount of volume displacement. And this is controlled with speed and time settings in the scripts that you execute, which, by the way, that is one of our benefits of this process. If you look at columns and liquid movement, there's always the potential for lack of consistency and lack of reproducibility. But in a process like this, you are executing a set of instructions that is actually the same every time you do it, whether it's today, tomorrow, next week, and so forth. And so now you have consistency and reproducibility in that entire process. Well, once all the mixing is done, the magnet simply goes back, we collect our beads, and then we repeat that process over. So as we go from sample through washes to elution, that's basically what we're doing. We let go of the beads, we mix, and then we collect. Let go of the beads, we mix, and we collect, and so forth. Now, the idea is that there's the potential to get better purity and yield in the process. Better purity because we're not moving those liquids around, just the beads, right? And so all those contaminating products end up being left behind. That means potentially better 260 to 80 ratios, for example. With the yield part, not only can we control the times and the speeds, but we can also control the bead collection process. So ultimately, we can make sure that we collect most, if not all of our beads throughout all of those steps. And if you're not losing those beads, you're not losing any of your product. It just makes sense. And this is the way that the KingFisher works. So if we keep it simple and we talk about the KingFisher Apex as being simply a magnetic bead mover, we can actually cover quite a few applications. So to break this down, we generally talk about it in two different categories. There's going to be your nucleic acid purification and then your protein purification. And with nucleic acids, that means being able to recover DNA, RNA, total nucleic acids. We can do it from a variety of samples such as FFPE, cells, tissues, blood, buccal swabs, and so forth. There's lots of kits for each of those. We can also do PCR cleanup, and we can do plasmid mini preps too, that we'll talk about a little bit later on as well. The thing with the kits is that they also tend to be validated protocols. So it's easy to open up the box and follow the instructions and get started, use the validated scripts, and you can pretty much get started right on day one. However, one of the advantages of the instrument is the fact that it comes with its own software. And on the Apex, that's actually on the touchscreen, so you don't even need a separate computer to do any of this. But having that software enables you to open up the instrument basically and make those modifications and customizations on those scripts to make the instrument work for you. So if you have to scale up or down, or you need to change that protocol because it's a unique sample type that requires those changes, it's all in your hands to be able to do that. On the other side, we've got the protein work. It's still magnetic bead-based chemistry, but what we do is we add a tag or an antibody to that bead and then basically go on a fishing expedition, right? And so it is a targeted protein approach and not a total protein approach. But if we keep that in mind as the concept, we've got a lot of different applications that we can cover. These are going to be your immunoprecipitations, or IP for short. Our co-IP, we can do different types of protein and antibody purification, phage display as a drug screening process. We can also do protein and peptide level cleanup for those mass spec analysis applications downstream. In addition to that, we can actually isolate things like exosomes and whole cells because they tend to express specific molecules that we can also target through that magnetic bead process. Now, ultimately, With protein work, it does tend to be more custom. And if you think about it, DNA is DNA, but there are a lot of different types of proteins, and so those workflows usually tend to be developed on the bench. Well, we can now use that software on the KingFisher Apex and basically write a script to mimic that process, and now we've simply adapted the workflow to the instrument to have the instrument work for you. So in the big picture, just keep that in mind. We can cover a lot of different types of nucleic acid and protein purification. So at Thermo Fisher Scientific, we also have a lot of chemistry for those applications, and it's always best to use our products first and foremost. Why? Well, we know our chemistry, we know the characteristics of the reagents and the beads, and it just makes it very easy for us to also help you make modifications if you need them, and also for troubleshooting if it ever comes up. And we've got a wide variety of kits for doing the DNA and RNA and total nucleic acid cleanup, working with proteins and cells and exosomes. Pretty much got every area covered. Those protocols are pre-made, so we do have the validated ones for the nucleic acid. We have starter protocols for proteins, but of course we can customize them as well. Ultimately, giving you the ability for faster processing with fewer manual steps. It's all optimized for automation. And when you put things onto the instrument, generally you tend to get better binding and washing efficiency. It's all streamlined. We can work with samples that might be a little more viscous. And we also don't have to worry about clogging those columns, right? Which can be problematic. So they are made for the KingFisher instrument. And of course, with the software, scalability, if you need to process more or less sample, it's really easy to go into the software to also make those changes. All right, we're going to go into a little tour of the KingFisher Apex. So I'm going to have the camera pan in a little bit, and while that's occurring, I just want to talk about accessing the instrument. So right now, we're already logged into the unit, but I want you to be aware that we have two different options for that. If you want open access on the instrument, once you turn the unit on, you're right away going to see your home screen like this, and anybody can use the instrument, anybody can do editing and creating protocols and so forth. But we also have an additional option where you can have restricted access. And here, anybody that wants to use the unit is going to have to log in with their credentials in order to access anything. And by logging in, you're also going to have 4 different classes of users. The first one is going to be your administrator, who has a lot of the administrative functions, of course, which is creating users and things like that. So they have full control of the instrument for the most part. The next class is going to be your scientist. They can do anything on the instrument. That means once they log in, they can create protocols, they can edit, they can run things, and there are no restrictions on that. The only thing they cannot do is add or alter their users, right? From there, you've got the personal admin. They can also log in and run protocols, but when it comes to actually editing, they can only edit protocols or scripts that they themselves have created. So they can use other scripts on the instrument, they just can't change them, right? And of course, the personal admin is not able to add or change the user inventory too. And lastly, we have the operator. Yeah, it's somebody you don't trust, but you need them to do the work. They're going to log into the instrument, they can pull up a script and run it, but that's pretty much going to be about it. And then lastly, one other comment too, if you do have credential login with passwords, if you happen to forget your password, we've also now have a password recovery system. You simply just need to contact us to get a code, and you'll be able to reset your password without losing everything. All right, well, let's kind of focus in on the home screen, because once you're in, this is pretty much what you are going to see. Large icons, everything is fully understandable and easy to navigate. If you want to run a protocol, you simply tap on Run Protocol. You'll go to the database, pull up your script, and it'll start executing right away. You can also simply open up a protocol, and that means pulling up the script, and you'll get to see the details without actually running anything. You can also create a protocol, and we're not going to go through the software tutorial at this time, but this, of course, is where you can create new protocols from scratch or basically do any kind of editing and modification that you want to do. In fact, I can usually give about a 30 to 40-minute tutorial on this, and you're pretty much an expert about 90% of the way to do your own work. Below that, just a list of recent scripts. They're usually listed in chronological order, but of course you can pin some ones at the top. And then we have a little side menu menu with some additional features as well, mostly for instrument mechanics, looking at reports and run history and so forth. But we're going to stay on the home page for the time being because I'm going to start a run just to show you that it's still easy to operate the instrument. So if you see your script on the home screen, you can actually tap on it and even open it up. And so if you see your protocol, you just tap the triangle, that'll go right away into the execution of that particular script. So you have two options. You can either tap the arrow and go straight to plate loading, or you can tap add lot information. And this allows you to input certain types of information for additional tracking. The first screen will be lot information. And really you can tap on each of these line items for all the different reagents. There's also one for the plate and the tip. In fact, you can also plug in a barcode scanner. So if you wanted to scan in unique barcode IDs for that plate, you can do that as well. Now, all of this information is optional. Additionally, at the very top, you've also got well notes, and this is where you can also input sample ID for everything too. So you've got your plate layout. All you need to do is simply tap on a space, and then you have three different options in order to add that information. One, you can use the touch screen and just basically type in whatever you need. Alternatively, you can have that barcode scanner and, of course, scan those items in, and that'll drop them into your plate map. Lastly, there's an import function at the top left or top right. And if you do that, you can import a CSV file that has all of that information. And you can modify or dictate how those samples go onto that plate map, either through rows or by columns. You can also dictate a starting position for where that information is actually input. But go ahead and put all this stuff in, and once you do, you can tap the check mark, and that'll actually go straight to the plate loading part. So as the camera backs out a little bit, we're going to get a better look at the inside of the instrument with the touch screen too. And so the touch screen is always going to tell you what plates to load. So there's no question about what we have to put onto the instrument. We'll start with the elution plate. It even shows you a picture of what plate type is supposed to be on there, reagent information. And so now we want to take our plates. I'm just going to grab a stack of them here that already have your reagent loaded into those plates. Now these plates also have barcodes on them. They have lot number information as well as the plate type, and we track all of this. But what you're going to do is take that elution plate with your reagent in there, put it into that front loading position so that the instrument can scan it. Now you'll see this change colors once we tell the instrument we've loaded it. And so you saw it turn green. That means it got the right kind of plate type that you were supposed to put into that position. It also moved to ask for the next plate, which is going to be a wash plate. And so just one at a time, we're going to be loading all of the plates in our script inventory right up front. And we've got the different flashes of green, each time showing us that we're putting the right plate in. So this gets us to the sample plate. The last one is going to be our tip plate. And this is simply going to be the tip cone we talked about earlier, sitting in a plate to present this to the instrument. Now the instrument will actually detect both of those barcodes, so make sure that they are facing the same direction. Once you're done, tell the instrument you've loaded it, it'll accept it, and then we go straight to the purification process. Now, the first thing it's got to do is pick up that tip comb, and there is a sensor for that. If it fails for any reason whatsoever, it will not continue with the rest of the protocol, ensuring that the magnet never touches any reagent, sample, and so forth. But if everything is great, we go straight to the sample plate and we start the processing. And you see right now we've got that empty tip comb. Basically just kind of sloshing our solution around, and this is the mixing process, right? Once we're done with all of that, the magnet goes back into that tip comb, and now it just slowly drops down in a couple of repeated motions, and this is going to collect all of those beads on the outer surface and tip of that tip comb. Well, when it finishes that process, of course, it's going to transfer to the next plate, and then we repeat that process as we talked about earlier, which is letting go of the beads, mixing, and then collecting the beads at the end. Now we're going to let this continue run. We're going to talk about some of the other features on the instrument. So at the end of this run, the instrument automatically generates PDF reports, and this is going to provide some tracking capability where you have a number of timestamps, tells you the firmware, the serial number, if you're logged in, who's logged into the instrument. It also gives information on the plates that you loaded and the reagents that you were supposed to put in there. Lastly, there's going to be an intra-run log. Everything is time and temperature stamped in multiple places, so you have a nice little record of the entire process. There's also an optional XML format in case you've integrated the unit. Now, as we're continuing with the process, let's go ahead and talk about some of the internal things. And my arms aren't quite that long, so I'm going to use a little helpful hand over here. What kind of features are enhanced, right? Well, we've got multiple heads. In fact, we can put up to 2 heads on the instrument now. So if you work with different volume formats, like a 96 and a 24, well, we've got them on there. And the 96 is very popular for general processing. The 24, as an example, has fewer number of samples, but it's about 5 times the amount of volume that you can process. Now, we also have 2 other types of magnets, and I'll just show them real quick to you. So we have a 96 combi head. Pins are a little bit thinner. They're round bottom. This is primarily for some lower volumes, but mostly to be able to go into storage tubes. In the past, for all of our KingFisher instruments, you had to elute into KingFisher plates. And then, of course, it's still an option. But if you want that additional functionality of going into either NUNC or matrix storage tubes, this is what's going to allow you to do that. And then we also have a 96 PCR head, right? Pins are even thinner and smaller. This works with 96 PCR plates, half skirted or skirted. And if you have to do those micro scale purifications where everything is 80 microliters or less, this might be the best option for you as well. But those are going to be your 4 head types that you can have on the instrument, with 2 of them being on there directly. Now, in addition to the 2 heads, we can also put 2 heat blocks into the instrument. And this is to either match the heads or match storage tubes and things like that. They are heat blocks, but they can actually heat up to 115 degrees and also cool down to about 4 degrees if you need that storage temperature at the very end. Additionally, there is a tip shield all the way in the back. You may have seen it. That actually goes underneath as we transfer from plate to plate. So this way, if anything falls off, it's going to land on there and not back onto the turntable. However, remember, this is a bead mover, not a liquid mover, and so the odds of that happening are pretty small. So I call it a secondary precaution, but it's nice to have on there. As we move around the instrument, we've also got some UV lights. There's 2, one on each side at the top, and this is to give you the ability to decontaminate your instrument. Now, we don't provide information on how. You have to establish your own protocols, but that means you can run that UV light after every run, once a day, once a week, however you choose to do that based on your material. You can also run it for 30 minutes or an hour, again, entirely up to you to establish those protocols. The barcode scanner, as you've seen in action when we loaded the plates, that is designed to scan the plastics barcodes, but also the accessories. So you may have seen the little barcode on the magnet, for example. As we assemble the unit, it just tells the unit what parts are installed and which position that they're in, so it knows how to move things around. And if you happen to be running a script that doesn't have the right parts, yeah, the instrument's going to tell you to put those on before you actually continue. We still have our 8-spot turntable. This is going to accommodate most applications that you can put on the instrument, but you're also unlimited, right? If you happen to have a 50-plate protocol, well, go ahead and program it. The instrument is just going to prompt you to swap things out periodically. We have the LED screen. That is going to be our front loading position. As you saw, it changes colors. The green acknowledges that you did something right. If it flashes red, it's an error, it's mad at you for some reason. But there are also some other color changes too to identify user input points or maybe different plate types if you code them into the script. Lastly is going to be the USB ports. We've got a couple in the front, some more in the back. And this, of course, is to attach that barcode scanner that we talked about, but also flash drives to import and export scripts or take reports off of the instrument. And for whatever reason, if you have an aversion to touchscreens, well, yeah, you can also plug in a mouse and a keyboard. And by the way, if you haven't noticed, we do have countdown timers on the screen. Tells you what step out of the total, the name of that step, and it'll even give you an actual temperature log, so you'll know where the temperature is and where it is supposed to be. Well, the run is completely done. Once this happens, you're simply going to unload all of your plates. And we'll just go ahead and remove those from the instrument, set them aside. All right, we're almost done here. And once the process is done and you have finished unloading all of your plates, the instrument will generate that report and you can exit that process and go back to the home screen if you would like to go ahead and do another run. The last thing I want to mention is the plastics. So we do sell KingFisher branded plastics. These are actually required to be used on the instrument. The main reason is shape, right? The shape of the tip comb, the shape of the bottom of the plate, the shape of the heat block that is on the instrument. They all match each other. And so it's important to do that. So number one, you don't damage the instrument, but it also contributes to the optimal processing overall. So please use the right kinds of plastics. We'll help you get these in your hands. All right, so that's it for our tour of the instrument. There are a lot more features that we can talk about if you reach out to us. But from here, we're going to move on to some recent application products that we've developed. Our first one is going to be the MagMax HMW, which stands for High Molecular Weight DNA Kit, right? And this actually piggybacks off of our popular DNA Ultra 2 kit that we've had out for a little while to do different types of biofluids and solid samples like your blood, buccal swab, cells and tissues, and so forth. Well, we've simply combined a booster solution to that kit, and this acts as a stabilizer to stabilize those longer fragments. So what the kit is designed for is to focus more on getting that DNA, longer fragment DNA for our long read sequencing needs. So if you need something at 100,000 kb or higher, this kit may be the right solution for you. And there are protocols that can accommodate various sample types, such as blood, cells, and tissues on a single run. So we have unified workflows for that. Lastly, it's also a very fast process, including plate setup and running on the instrument. We're talking about getting those results back in less than 2 hours with no overnight process. All right, the next kit is going to be our MagMax Sequential DNA RNA Kit. And this is a nice kit to have because in the past, if you have very limited precious sample, what you really had to do is take that sample and split it. Well, right off the bat, you're losing 50% of your sample. And so this one is designed to process that same sample in a DNA run to get your DNA, and then you're going to put it through a second run or take that sample plate in the second run to capture your RNA. No more splitting samples, and you get both DNA and then RNA in that sequential process. Now, right now, the kit is validated for blood and bone marrow. We're looking at adding other sample types to expand on that. But at least to start, this is going to be great for the hematological testing to get both of those nucleic acid types in that purification process. All right, our next one is going to be our MagMax cell-free DNA. Now, this is not necessarily a newer kit. It's actually been around for a while, but it's designed specifically for areas like cancer diagnostics. In the past, what we used to do is take somebody through surgery, right? And that would be a very invasive way to collect tissues and tumors and things so that they can be analyzed. Well, it also makes it difficult to go back and get another sample if that happens to be the case. So this is a method called liquid biopsy, and that's what this kit is designed to do, is to really provide a non-invasive approach where you can collect that blood sample, isolate the plasma from that, and then focus on enriching for the cell-free DNA component of that. Because basically when tumor cells start to die in apoptose inside a person, they're going to release all that genetic content, and those smaller fragments can really tell you more information about the type of tumor, stage, and so forth. And so the kit focuses on isolating those smaller fragments so you don't have to worry about that genomic DNA contamination. That might make it a little more difficult to do that. The other thing too is flexible input volume. So we have a lot of validated scripts already. So whether you want to do a few hundred microliters or a mil, 2 mil, 4 mil, and so forth, we have all those available. And in fact, since we can customize, we can even help you do something in between if needed. So wide variety and flexible input volume. Lastly is going to be our MagMax Pro HT no-spin plasmid mini prep kit. This also came out earlier this year, and I really love this kit because of its ease of use and really streamlining that mini prep process. As you know, if you've done mini preps in the past, it's about growing up that culture. You lyse, you neutralize, right? You've got to clarify that lysate before you can really put it into any type of purification process. Well, this kit uses a two-bead system. So what you do is you start straight from culture. You put that culture into the plate with that first set of beads. The bacteria is going to bind those beads, and we do the lysis and neutralization and everything on the instrument itself. Well, we're going to pull out that first set of beads, and that is the equivalent of clarifying our lysate. There'll be a pause point. We'll add a second set of beads and some endotoxin removal reagent, and this is now going to allow you to bind, wash, and purify. Amazingly, all of that from culture to elution at the end with purified plasmid is about 35 minutes or less to be able to do that entire workflow. It is all centrifugation free since we do it all on the instrument, so you don't have to worry about the extra equipment and the processing steps. And lastly, I mentioned the endotoxin removal reagent, so it is transfection ready. That means getting less than one endotoxin unit per microgram in your results. And finally, high quality and yield. Now, depending on your input, the plasmid size and the media type and culture density, there is the potential to get up to 20 micrograms of plasmid DNA in each of your samples. So you'll have plenty of material to do that screening or any downstream application that you need after this. So from here, we're going to shift to our protein applications. Now, we won't be able to cover everything in detail, but understand Thermo Fisher Scientific has a large number of magnetic bead-based offerings for protein purification based on ligand-mediated binding. We can categorize these products into four main groups. They're going to be protein immobilization. That's where we have functional groups such as our amines, sulfhydryls, carboxylic acids, aldehydes, tosyls, and even epoxy for covalent attachment. The second group is going to be our antibody purification-based beads, and this is where we use protein A, protein G, the combination of protein AG, or even protein L to capture those antibodies. We have biotin tag capture with a variety of avidins. The most common and most popular, of course, is our streptavidin, but you can work with regular avidin and neutroavidin as well. And finally, the fourth group is going to be our recombinant tag purification. And this includes our nickel cobalt NTA, our glutathione, and a variety of anti-tag versions such as anti-MIC or flag tag and so forth. Now, these tools enable targeted isolations of proteins through specific biochemical interactions. They're primarily available through our Dynabeads and our Pierce brands, which are fully automatable on the KingFisher platform. So let's just take a little closer look at each of these. We'll start with Dynabeads. Now, these are our super paramagnetic uniform polystyrene particles widely utilized in protein purification techniques, such as our IP and co-IP that we talked about. They can be used for affinity purification of his-tagged and antibody-bound proteins. They can also be used for protein-protein interaction studies. Now, the beads enable gentle, efficient separation of target from complex lysates. No need for centrifugation because it is all done on the instrument such as the KingFisher Apex through high throughput automation. A distinct advantage? Well, the non-porous defined surface chemistry, which minimizes non-specific binding and helps prevent the trapping of unwanted proteins within those internal pores. Ultimately, this results in yields that are of high purity, higher yields overall, and reduced background noise in downstream analysis. Dynabeads have been a gold standard for a long time. And while Dynabeads can be used to conjugate your own antibodies for any target, there are also a wide variety of products we have available to save you time that are pre-conjugated to target some common proteins like different CD molecules, for example. So the other type are going to be our Pierce beads, and there's two different classes for this. The first of all, the Pierce magnetic beads are also versatile superparamagnetic particles, and they're designed for their own variety of protein purification workflows too. These include immunoprecipitation and co-IP as well, so there is some overlap in those applications, but they target antigens from cell lysates or tissue extracts. They can be used for the affinity purification of antibodies and the high capacity isolation of his tag recombinant proteins using things like our nickel IMAC variants. They're all great for studying protein-protein or protein DNA interactions, and of course, all are compatible with automated systems like our KingFisher instruments. That was the first one, which is our recombinant protein purification using the IMAC beads or the use of a variety of anti-tags as displayed in our slide here. The distinct advantage overall is their nearly 4 times higher binding capacity. And so this allows researchers to use smaller bead volumes per experiment. It also reduces costs, minimizes the need for sample dilution, and enhances overall efficiency. And it does this while maintaining low nonspecific binding through stable pre-blocking. Now, the second category is antibody bead-based purification. And here you can conjugate your own antibody to the bead to capture your target of interest. These, of course, are going to be our protein A and G varieties, protein L, and of course, streptavidin, as we talked about earlier too. And if you're really looking for high binding capacity, well, look for our Pierce magnetic agarose beads for that bulk purification. So we've covered quite a bit to showcase our KingFisher Apex, how it works, what it can do. We've also featured a handful of magnetic bead-based chemistry and products. However, I want you to know that these are really only a subset of all the products that we have available in terms of nucleic acid purification, and there are a lot of specifics about protein purification as well. I encourage you to reach out as we have a number of resources to help guide you to the right fit for your needs. In fact, we even do instrument demos in person, so you can, kick the tires, see how the instrument works, and run some of your samples with us so we can help you get started right away. We're going to go into the Q&A. If you have any questions, go ahead and put those into the chat right now, and we'll see if we can answer some.
Hey, quick question about sample types. I was looking through some of the kits and protocols that were available. We work with some pretty unique sample types. If that's not available in your protocols, is that something we can still work with you guys on?
Yeah, absolutely. So I mentioned earlier, we've got a lot of kits to cover a variety of areas with nucleic acid purification, but sometimes it's just impossible to cover everything. Well, we have field specialists or field scientists that can help with that too. And sometimes it just means going into looking at your sample type, looking at our available kits, and thinking about what we can do to modify some of those kits to fit your needs. And if it comes down to where you are also creating your custom home brew, well, remember, we've got the touchscreen and the software on there, so we can help you create that script to do that process. But we prefer to start with our kits since we're usually most of the way there, and we can usually help adapt these custom or unique sample types onto the KingFisher. So absolutely, we can do that, and we'll make that as part of the demo too.
Perfect, thank you.
Mm-hmm. All right, well, I think that might be all for the questions for today. I appreciate the thought-provoking dialogue here. If there's anything else that comes up, feel free to reach out to us. But we're going to go ahead and wrap this up, and my mic drop moment. See y'all later.